All right, it's time to start our Structure Meets Function webinar. This is number 41, and we have today two speakers, both who have done work at the University of Helsinki. I'm Hanna Oksanen, also from the University of Helsinki, and I'm the coordinator of Instruct Finland activities in Finland, national and also the international operations. So what is the Instruct-ERIC? Oh no, I can't change the slide for some reason. Now it works. So Instruct-ERIC is a research infrastructure which operates in Europe, and Instruct have 17 members, of which most of are hosting this kind of Instruct centers, which are supporting this kind of a high quality research. Instruct-ERIC is providing this kind of access to work on these service units, and also training courses and internship, and also funding for R&D. What is coming next spring is the biennial structural biology conference. This will be in Brussels, Belgium in May, and the registration is already open, and there is a possibility to submit abstracts for oral presentations. There is also students fellowship available, and rates for lower accommodation. So take a look on the website, and if there are sponsors who would like to support our event, that's also highly welcome. The Instruct-ERIC website is there for your way to find all this information, and also there is a support email address that you can ask for help. Let's continue with Joseph. So Joseph is from University of Madrid, Universidad Autónoma de Madrid, and he worked also in Helsinki, and his work was supported by Instruct-ERIC, but also by this ISIDORe funding consortium. So he will talk about Monkeypox virus. So please, floor is yours.
Okay, so thanks a lot, Hannah, for the introduction. I hope you can all see the screen well. Okay. Well, as Hannah said, my name is Joseph Patrick McGrail. I'm a fourth year PhD student belonging to Dr. Susana Guerra's lab at the Autonomous University of Madrid, here to give you a brief overview of all the results that were possible through our collaboration with the University of Helsinki, through Instruct-ERIC and the ISIDORe consortium. So this talk will be split in two main parts, with the first one titled Comparative Analysis of 2022 Outbreak Mpox Virus and Previous Clade 2 Strains. Now, first off, to give you a brief overview of the pox virus cycle, what I'm going to say is that pox viruses are extremely large viruses with a double-stranded linear DNA of approximately 190 kilobases. This allows it to express over 200 viral proteins that can be split between early, intermediate, and late. This also allows it to have all the machinery necessary to replicate in the cytoplasm. So it also has a complex morphogenetical process that I'll explain to you with the help of this scheme that you can see here. So first off, the virus enters the cell where it undergoes uncoating and then starts forming early proteins. Now, these early proteins will inhibit the immune response and help in DNA replication. Then it will form intermediate and late proteins that will start helping form the actual structure of the virus, becoming immature viruses, which then lead to the first main viral form, MVs, or mature virus. Now, mature viruses, MVs, are more linked to localized infection and person-to-person transmission. And then this MV undergoes wrapping through the Golgi apparatus, acquires a double membrane, And then once it has this, it exits the cell and turns into the second main viral form, EV, or extracellular enveloped virus. And with this viral form being more linked to cell spread. So MVs are more from person to person, while EVs are more within the actual individual. So after this, I'm going to give you also a recap on the pathology in humans caused by poxviruses. So smallpox will ring a bell as one of the most destructive infectious diseases in all of mankind. It has killed millions throughout human history, but happily, successfully eradicated by the World Health Organization in 1980 through a successful vaccination campaign, which marks one of the biggest possible successes in public health history. So after this, the poxviruses weren't in the spotlight for quite a while until the 2022 global outbreak of Mpox, which for the first time, while this disease was only normally like individual cases around Africa and some in the USA in 2003, it had never become at such a wide scale and had this increased human-to-human transmissibility that differentiated from other previous older strains. Now, while the disease of Mpox in itself is not as severe, causing just pustules, painful pustules across your body, it is had a wide array of number of co-infections with HIV-infected individuals, which led to quite a severe variant of the disease that had a very high mortality. And in any case, we have to keep an eye on this disease because it could occupy the niche that smallpox once did. Although for now, it is not as deadly. We hope it stays the same, but it is right now being transmitted across much more humans with actual community transmission than before. So when it comes to the phylogenetics of it, Mpox virus is subdivided into clades 1A, 1B, 2A, and 2B, with clade 1 causing more severe symptoms and higher mortality located primarily in Central Africa, while clade 2 produces lesser symptoms and is located mainly in Western Africa. Now, the 2022 outbreak strain belongs to clade 2B, deriving from clade 2, and its main characteristic was its much higher transmissibility, though now the 2024 DRC outbreak Mpox virus and all the cases that you have been seeing more in the news belong to clade 1B, with this now also acquiring an increased human-to-human transmissibility, being quite worrisome. So this has caused a huge number of cases that you can see in the map all worldwide, leading to nearly 170,000 cases, 441 fatalities, and affecting over 140 countries. So the main question is, what has changed? What has made these strains be more transmissible from humans to humans than previous strains? One hypothesis that our lab believes in also is that the difference in 2022 Mpox transmission due to a change in the viral morphogenesis and the amount of infectious form production. Is the MV-EV ratio different? Much more MVs helping more localized lesions and more person-to-person transmission rather than EVs that could sustain within the individual. So this is what we want to check out. To do this, we have the following objectives. We're going to characterize the replication dynamic of the 2022 Mpox in comparison to previous clade 2 strains, USA 2003, which was cause of the Wisconsin outbreak in 2003, that did not present any human-to-human transmission. It was zoonotic in its nature, and Rabies, which is our oldest reference isolate that was obtained from infected cynomolgus monkeys. All this has been done in mouse embryonic fibroblast cells, MEF cells. Why MEF cells? Because rodents are hypothesized to be one of the main reservoirs of Mpox virus. So it is a quite important system to study, not just human cells, but also probably the host cells. So we're also going to compare molecularly the protein patterns produced in each strain infection to discover differentially expressed viral or cellular proteins, which is where our friends at the University of Helsinki helped quite a lot. So first off, in this result image, you can just see a standard plaque assay. We quantified intracellular titer as a way of trying to measure MBs and extracellular titer as a way to quantify EVs. What you can see is that the 2022 strain goes much slower than the other previous clade 2 strains. And when we look at EVs, the USA 2003, the extracellular titer, is much higher than both strains, suggesting that this strain had much more EVs, while in 2022 does not produce as much. This is supported by the plaque phenotypes that you can see on the right, where when we did the supernatant dilution, you can see comet-shaped plaques, which are normally associated with much more EV production. And when we look at intracellular dilution, you can see that 2022 has very, very, very small plaques, while USA are much larger. While So this helps suggest the fact that maybe 2022 does not produce as much EVs and much more MVs. Then we just complement this with immunofluorescence. So you could see that there's much more infection in USA 2003 and rare compared to 2022, which just goes very slowly. And then we did a Western blotting analysis using different vaccinia virus protein antibodies, which, since it's the poxvirus reference strain, it has a high homology with Mpox and can help us along to actually visualize these proteins. You can see that once again, 2022 does not have as much protein amount compared to the others. And even F13, which is a protein linked to EVs, is in a much, much higher amount in USA 2003. Once again, supporting that this strain has much more EV production while 2022 does not. And Ryer is just intermediate. So the conclusion from this section is that 2022 has much more slower viral cycle and that USA produces much more extracellular associated titer, making a big difference between the evolution of these two strains. Now we move on to our proteomic results of this section. What we did is we did a proteomic analysis of infected cell lysates at an MOI of 21 hours post-infection with all three strains. And what you can see on the left is a heat map of all the differentially expressed proteins. We were comparing log-to-fold changes of the infected versus the mock. You can see that once again, USA has much more higher amounts of abundance of viral protein compared to 2022. Though, and then on the right, you can just see a volcano plot, just another way of visualizing that once again, many, many more viral proteins produced in USA 2003, much more abundance. Though we did find 3 viral proteins that were higher in 2022 compared to USA 2003, which were J2 thymidine kinase, O1, and K1, suggesting that maybe these proteins could have had a role in this increased human-to-human transmissibility, though this needs to be confirmed. Then we looked at the biological process pathways, the enrichment, which pathways were more altered in all 3 strains. And what we saw is that 2022 Mpox was actually the strain that caused most process enrichment, fold enrichment, in processes such as supramolecular fiber organization, actin filament-based processes, and cytoskeleton organization, which makes sense because poxviruses use our cytoskeleton to their advantage, using actin tails to catapult themselves. And it does seem that the 2022 strain is using these processes better, or it's causing much more alteration in these processes than the other 2 strains. So, as mentioned before, these processes are the most enriched pathways in the infection, but the fold enrichment is higher in 2022. So, to conclude this section, 2022 Mpox produces a reduced viral growth compared to the other strains. USA seems to produce much more extracellular viral titer and more EV-associated protein expression. Proteomic analysis displays that J2, O1, and K1 are the only viral proteins that were more elevated in 2022 compared to USA, and that's fiber and actin cytoskeleton organization were the most enriched pathways in Mpox infection, with 2022 causing highest fold enrichment. Now I'll move on to the next section. Hopefully I'm not bad on time. And what we can say is that now a big question also, another hypothesis that for why Mpox could have caused an increased human-to-human transmissibility are host factors, with ApoBec3, for example, being known to edit the actual genome of the virus in an effort to stop infection, but may have ended up causing mutations that have helped its increased human-to-human transmissibility. So with host factors being a point of study, our lab is a reference in the study of ISG15 and in poxviruses. So what we're going to do, we are going to see ISG15's role in Mpox virus infection. Now, what is ISG15? ISG15 is interferon stimulate gene 15, one of the most abundantly produced ISGs, with its main characteristic being that it is a ubiquitin-like protein that can bind to de novo synthesized proteins in a process called isolation. Now, isolation is known to be highly antiviral, and it can hamper the growth in influenza and in SARS-CoV-2, with these viruses having developed mechanisms to counteract isolation so it does not stop its viral growth. And also IC15 has an extracellular form, so it can have immunomodulatory activity and activate cytokines. So it is a very interesting protein to see in the context of Mpox virus infection. With our previous work demonstrating that IC15 is important in cancer, exosome regulation, macrophage and mitochondrial metabolism, and as a potential vaccine adjuvant. With even recently we haven't found that IC15 was found to have a role in regulating the dissemination of the AIDS-DJ vaccinia strain in MEF cells. So we want to see what is going on in Mpox. So for this, we have the following objectives. We're going to characterize the replication dynamic of the 2024 Congo outbreak strain, which we were very lucky to have been able to access, gain an actual clinical isolate of this new and very relevant strain. So we added it with a pool of our other strains, and we did characterize it in wild type and IC15 knockout MEF cells. We also performed a proteomic and phosphoproteomic analysis where we made use of the Instruct-ERIC infrastructure, and to understand how IC15 absence influences protein production and phosphorylation. If this skips. Okay, so once again, we did plaque assays. We want to see how it grows with and without IC15. You can see that in general, there is an increase, but in only one strain, this increase was very notable, which was in the Rabies, the oldest reference strain, while the newest did not seem to present as much of an increase, with especially the 2024 and 2022 not having as much of an increase. So we complement this with immunofluorescence just to see the amount of infection. Once again, there are more infected cells, more green in the knockout, though in 2024, for example, it was not as notable. Then we did a Western blotting analysis. We compared the clade 2 strains, the clade 1b versus 2b also, which is on the right. And what we saw is that you can see is that there's always more viral protein in the knockout compared to the wild type. In the densitometry analysis conducted below is just another way of reinforcing this after 3 replicates. So what we can say is that viral progression of Mpox virus is accelerated in the absence of IC15. And then we also wanted to visualize the viral forms that we were so much interested in. So what we did is, with help of the Eurobioimaging infrastructure also, that was helped along by the ISIDORe consortium, we looked and saw that there was a higher amount of intracellular viral forms within the knockout infected cells. This was seen in 2022, USA. And when we looked at this, I had to basically go over many cell profiles, identifying the different viral forms. You can see the immature virus that turns into the mature virus, undergoes wrapping, and then just becomes a wrapped virus. So I had to count and quantify. And what we saw is that there is an increase in the accumulation of intracellular viral forms. So this seems to be going along with the results that we saw before. Now it's full proteomic from here. No more microscopy. So now what we did is, once again, with the help of the University of Helsinki and Markus Varjosalo's group, we obtained heat maps and we fold changes that when we compared the knockout infection versus the wild type infection. And here we look specifically at viral proteins. And what you can see is that in each strain, in general, there is a higher amount of viral protein in the knockout infection. Though what is also notable is that Rabies seems to be the strain that has much more increase of different viral proteins, and that in the clade 2 strains, now 2024, there is a reduction at the extreme ends of the genome, since this heat map is ordered by OPG, and the ends of the genome are more linked to inhibiting the immune response. So in clade 2, there is more of these proteins in wild type than in knockout, but this phenotype is not seen in clade 1b, which makes another difference between this 2024 and these clade 2 strains, suggesting evolutionary differences. But what we can say is that IC15 seems to regulate the viral proteome. Now, looking at the actual host proteins, we also found a shocking result that when I saw it was quite, wow. Basically what happened was that when we looked at comparing knockout versus wild type, normally there was quite a high alteration of the biological processes in the clade 2 strains. And when we did the 2024 strain, what we found is that while there was a higher amount of viral protein in knockout, there was not as much of a difference in host protein alterations between clade 1b 2024 infection between wild type and knockout, which you can see visually in this figure that just the green balls of the network graphs are much smaller because there were much less proteins in 2024 compared to the other ones, and same in the PPI interactions. So it's quite shocking. It says that seems the IC15 absence does not alter significantly clade 1b host protein expression. There's not as much of a difference between wild type and knockout. This does not mean that there is no alterations. It's just that between the wild type and knockout, there's not as much of a difference. And lastly, we move on to phosphoproteomics, where we want to see if there are any differences in phosphorylation when IC15 is absent, since there have been previous studies that have suggested it has a role in regulating phosphorylation in, for example, COVID infection. And what we saw, once again, is differences between clade 1b and the clade 2 strains. There is a drastic reduction in phosphosites and a limited reduction in phosphoproteins in the clade 2 strains, which are 2022 USA and rare. But when it comes to the clade 1b 2024, there is not as much of a difference. And you can see this also in the volcano plot where the pattern is more equal in the clade 1b, while in clade 2, you can see that there's always seems to be more phosphorylated, the differentially phosphorylated proteins in the wild type compared to the knockout. While this was only more significant in USA, the pattern, even though it did not reach the cutoff values, you can still see them that it follows this pattern. And when we looked at phosphorylation, altered phosphorylation pathways, we saw some interesting ones like nedylation, mTOR, or single transduction, that even though the clade 1b does not have as much of a difference between wild type and knockout, there were still quite a few interesting pathways. So what we see is IC15 absence dampens phosphorylation, but only in clade 2 mpox infection, not in clade 1b. So to conclude, I just want with this image is that basically what we have found in this section is that there seems to be evolutionary differences between how this strain reacts to IC15 absence of clade 1b, with it maybe having mechanisms that counteract IC15's presence in the first place, so making it that if it's absent, it does not alter much the course of the infection. With the 2024 having a similar phosphorylation, only slightly altered host processes, but still an increased protein and virus accumulation. While in the clade 2 strains, the differences are much more notable. And I just want to give just one final thanks to my lab, to my PI Susana Guerra for allowing me to give this presentation, to all our members, to our collaborators at the EMBL, Mount Sinai CMB, to the ISIDORe consortium for helping fund the project that was given to my PI, Eurobiogen, of course, but especially to Instruct for allowing me to give this webinar, and to the High Life Institute, University of Helsinki, Marcus Variosalo, and Titukala Salakaskitalo, for helping along with the proteomic part, which is a very important part of all this process. And yeah, so thanks. It's not appearing. Well, it's supposed to say thanks for your attention. Any questions? But yeah, that's it.
All right. Thank you, Joseph. Very nice presentation. A lot of data. Did you collect this all proteomic data here in Helsinki, or was it some part done in your lab? Do you have this kind of a proteomic facility there?
No, we basically just infected the samples at the BSL-3 in the Instituto de Salud Carlos III, and the whole the processing and the data analysis was done in the University of Helsinki. I was not there, it was just sent there, and they were the ones that did everything.
Yeah.
So it's big help.
Yeah. Yeah, it looks like that. It helped a lot. Yes.
Yeah.
And this is also one very good that you can use these services remotely as well. That you don't need to travel. The samples can travel. Of course,
exactly.
if you would be on site, you can feel the different environment, but it is very nice the way to do it. So please, you can put your questions to the Q&A chat for Joseph. So I can ask though. So in your first part of the talk, you mentioned these 3 proteins which were, if I got it right, that they had a higher expression in this 2022 strain and somehow changed the human to human transition. Have you followed that line somehow, and do you have more understanding what's there?
It was something that I wanted to follow through more on. We haven't, but basically the functions of these proteins, J2, as a thymidine kinase, is to help the DNA replication along. K1 is inhibiting the immune response, and O1 is not as understood from what I've gathered in through the publications. But the problem is there is also another explanation that is that these are more or less early proteins. And since 2022 is going slower, that there is a point that it could be that the reason they're more abundant is that because USA is just going more quicker. It could be that. So that's the, like, it would be nice if it was more due to the human transmissibility, but it could be also that. And the way to maybe follow through is that would be to maybe like insert these proteins in vaccinia virus, which is level 2 pathogen. But that is quite a bit of work. So we haven't really had the time to do it, but we would actually more or less like to study these proteins in the future.
Yeah. Sounds interesting. This also, I'm just virologist, so those were lovely images of those through the virus life cycles that you take with the Eurobi imaging. So that work was not done in Finland, so it was somewhere else.
Yes, no, that was in EMBL Heidelberg. No, it was good, but it was all thanks like the ISIDORe consortium. Oh, that was a good part of it. That also helped us gain that structural biology edge through the University of Helsinki, but also the microscopy facilities. But there I was not for the actual images, which were all done by Viola Orshot there, but I was there present. I managed to go there, but I couldn't go to the 2 places. I really wanted to go to Helsinki though. Yeah, it would have been nice.
Yeah, how you quantify those intercellular virus particles? Of course, you can use the images, but it's always like a bias. Do you have some kind of a mechanism?
Yeah, no, definitely. Like this was even something that Viola even said that when it comes to cell profiles, it's always a bit risky because obviously you're just getting one slice of a whole cell.
It's
there's always a problem that's always going to be a bit of a bias. We try to quantify as many as possible to love cell profiles to try to reduce this bias, but this experiment is more like complementary to what we've seen before. Obviously, if it was the only thing, when we measure the intracellular virus, we're seeing that there is a higher amount just by plaque assay. So, but it is a problem to only rely on this because, as you say, it is a bias, and also it's all by eye. And sometimes these viral forms are not as easy to spot, especially the wrapping process. That's hard.
Yeah, and also depending on that how they are if you are seeing those particles on the top, they might
look different.
Yeah.
That's why in the graph we say we don't say like by the cell, we just say by cell profile, and it's definitely a limitation.
Yes. All right. So any questions from the audience? Now it's your chance to ask questions from Joseph, technical or scientific, or if you're interested in any other topic. All right. So if no further questions, so let's thank Joseph again, and I thank the audience for listening, and please enjoy the next webinar. I can't remember when it will be, but there will be one more webinar coming soon, and then the biannual meeting is in May, so please take a look on the website and register. All right, hopefully we will see you there. All right, have a nice day.
Same.
Thank you, Hannah. Thank you, everyone.